I'm Hani. I hail from UCSF, where we believe you can't have too many affiliations, and these are some of mine. I'm actually moving my lab down to the ARC Institute in a few months, so there will be a reshuffling of these. So my lab is actually kind of a technology-first lab. We're systems biologists. I trained as a computational biologist back in the day. I slowly moved towards experimental biology in cancer, and kind of my lab is at the intersection of gene expression control, especially focused on RNA biology, cancer biology, and computational biology. And our lab is about 20-25 people, half dry, half wet, kind of working together, both in diagnostics and therapeutic areas. But my lab predominantly is focused on metastasis and cancer progression. Actually, when you think about the field of cancer, there's a lot of focus on tumorigenesis, but the metastasis field is actually not as large. So kind of a smaller subfield is really dedicated to this for a variety of reasons, just kind of ease of access to models and samples being a big contributor to this. Then I'll touch on some of that and why this platform can be very helpful. But also, at the same time, it's kind of the major cause of death and mortality from cancer. So it's an area that continues to be underexplored, and that's where we are most focused on. Now, our approach to studying metastasis is really we view it as a disease of disordered gene expression. So the idea being that there are regulatory programs in every cell that decides what genes are expressed, when they're expressed, and to what extent. And cancer cells are basically hijacking different components of these machineries and then using them to kind of dial up or down different genes and pathways. So that I'm anthropomorphizing the entire process, but the idea is that cancer cells that can actually do these kinds of achieve these kinds of dysregulations will grow faster, grow better, survive longer, and ultimately metastasize to other organs. And of course, you can actually see this kind of reprogramming of the gene expression landscape in clinical samples as well. So in the context of metastasis, actually having paired samples is quite rare because often there's years apart between the primary and met sample collection, sometimes at different institutes even. But there are a few studies done over the years where there's a matched primary and met, and you can actually compare them quite effectively. Of course, there is some confounding by treatment as well. So basically a lot of the mets are treatment resistant, whereas the primaries are treatment naive. But predominantly what you do see is a picture like the one I'm showing on the right. It's basically a gene expression landscape of, as you see, thousands of genes are just different between the primaries and mets. And what is important is that a lot of this is actually not driven by mutations. So mutational drivers of metastasis are actually quite rare. And this is, again, very much distinct from tumorigenesis, which is really driven by a few clonal mutations in the beginning of the tumor's life cycle. Now, as I mentioned, we are really pretty much interested in kind of what else goes wrong. If it's not mutations, then there are all these epigenetic and other regulatory kind of pathways that are perturbed in the context of metastasis. We have been specifically interested in post-transcriptional regulation over the years, partly because there's just a lot more to find there. So there are just more lower hanging fruits, and we go where the data takes us, and more we have actually predominantly gravitated towards post-transcriptional regulatory processes. And this is basically anything that happens to RNA after it's transcribed. Once you get from a pre-mRNA, you get processing, and you get export, you get degradation or translation if it's an mRNA. And each of these steps in a normal cell is like tightly regulated. So there are kind of regulatory programs sitting on top of every single one of these arrows. And what we see in the context of cancer is that, and cancer progression, is that every single one of these arrows are also impacted. And a lot of them are actually having driving roles in cancer progression. And I've cited some of the work that we have done for each of these steps basically over the years, going back to my days as a postdoc, where a lot of it also very recently in the lab. Now, I mentioned that studying metastasis is hard. One of the reasons is that it's a really complex cascade of events. So, in order to have a cancer cell that successfully metastasizes, you need to basically get from the tumor into the bloodstream, the process called intravasation. You need to survive in the bloodstream. Then you need to get out of the bloodstream into your distal organ, and then survive in that new environment, escape dormancy, and basically recolonize and grow a secondary tumor. So this is a very complex process, and every bit of it requires different talents from cancer cells, and which is why to complete this cascade, a lot of things have to go right for the cancer and wrong for us, and that's partly why metastasis is kind of rare. And different parts of this cascade also have different rates of success. For example, these earlier steps are easier, so that's why you get a lot of circulating tumor cells, for example. Which I assume Deepcell has been looking at as well. So the reason is that the intravasation step is actually easier. So even in earlier stage tumor, you do have cells that find their way into the bloodstream, but for the vast majority of cells, they will just simply die in the bloodstream. They don't survive for long. A very small percentage would actually manage to get to a distal organ and survive in that distal organ. And in that population, again, the vast majority of them will remain dormant. And sometimes decades later, one of them can switch out and grow a new tumor. And that process, again, is just something that we are, as a field, studying. Now, the take-home message from this cascade is that this is actually impossible to recapitulate in vitro. So that's one of the reasons that the metastasis bit has been held back, is that it's very difficult to model this process in a scalable way. So we are very much limited to mouse models. And while we have built assays for each of these steps, they're, again, fairly limiting. For example, these are famous invasion assays. So the idea is that cancer cells that metastasize better tend to invade better. So you can actually, if you have a driver of metastasis, and if you want to know which of these steps are impacted by it, you can go and do those assays. The difficulty is that the opposite doesn't work. So if you do a screen for cancer cell invasion, most of those factors that come out of it will not be driving metastasis. So things that drive metastasis impact invasion, but not vice versa. So as a result of all these complications, it's a field that has really been held back by model systems and way of studying them. Now, with that introduction, let me tell you a little bit about how we actually do end up studying metastasis in the field. And that's basically one of the ways that we do this is these in vivo selection models. So this is basically an idea that was put forth by Josh Fiedler back in the 70s, who did this for the first time, and later on was championed by John Massac's group at Memorial Sloan Kettering. And now I think since then, so he did it I think early 2000s, and since then us and many other labs have adopted this strategy. I think there are 50 or so of these models now in different labs. So the idea is that you start with an established cancer cell line or PDX or organoid, and these cells are human-derived. They have a baseline metastasis, and by baseline I mean that if you put it in immunocompromised mice, in the primary site, so for example, we do a fair bit of breast cancer, so you can inject them into mammary fat pad of mice and grow a breast tumor, which in a xenograft setting, and then watch them metastasize. And from breast, for example, you tend to go to lung or brain or bone. So let's say we are looking at lung. If you take the metastatic foci out of this mouse and re-inject it into a new mouse, you actually see that they're much better at metastasis in the same mouse model. So after a few rounds of selection, you kind of plateau, and that's where when you stop. It's usually around second or third generation, sometimes fourth. But what you end up having is that basically a highly metastatic derivative of your parental population that is orders of magnitude more metastatic. So this is one example I'm showing you here. This is a long model of breast cancer. The parental line is a model of triple-negative breast cancer. It's called MDA-MB231. And there is a highly metastatic derivative of it, which is called MDA-LM2. This was done in John Massac's group at Memorial, as I mentioned. And this model is basically generated after 2 rounds of selection in mice, which is what that 2 stands for. And as you can see, the metastatic burden in mice is orders of magnitude higher. These lines are otherwise isogenic. We have actually done whole genome sequencing, exome sequencing. There is very little that changes in terms of genetics, but as you can see, the phenotype is vastly different. Now, even from those early days, folks in the Massager lab themselves, they did microarrays to look at gene expression changes, comparing parental cells to highly metastatic derivatives, and they identified genes that are up or down regulated. And this was really the first instance that they started knocking down these genes and showing that if you knock these genes down individually, you can actually get lower metastasis. So this is the control line, and these are the genes that they tried. And as you can see, each of them that you knock down, you get a reduction in metastatic potential. And this was published 2 decades ago. Now, what is important here is that we are, again, looking at this population as kind of this bulk idea, right? That there's a cell, they all look like the same, they're parental, and then there are these highly metastatic derivative, they're also separate. In reality, these cells are kind of hodgepodge of variants. They were collected, a lot of them, in a heterogeneous way from patients anyway. So the NDA cells, for example, they came from pleural effusion. They were actually not a single clone, never. So it's a heterogeneous population, and that heterogeneity is intact. If you look at, and this was done quite some time ago, that pre-Deepcell era, PDE. And basically, this is just normal microscopy, and you can see there's a fair bit of variations in terms of different morphological features. And if you look at the comparison of the parental line versus highly metastatic line, the confidence intervals largely overlap. You actually cannot tell the difference for the vast majority of these lines, going from poorly metastatic cell to highly metastatic cell with these simple features, and certainly not with eyes. And that's actually problematic for us because we have a ton of these lines floating around, and the parental and the highly metastatic look identical on the microscope. So you don't know actually you have the right line until you go back and put it in the mice and measure metastasis. So as you can imagine, contaminations and mislabeling are a headache for us. But one of the things that do work, as I mentioned, is RNA sequencing. And of course, you can also look at it, capturing this heterogeneity. You can look at it with single-cell sequencing as well. This is a comparison of parental and highly metastatic derivatives for one of our models. This is another model of breast cancer metastasis that we have generated in our lab. The purple ones are the parental ones, and the green ones are the highly metastatic ones. And this is kind of endogenously controlled. They were in the same they were in a mixed population, barcoded separately, so they're genetically barcoded. So this difference is really not batching of samples or anything like that. But as you can see, there is a fair bit of difference in the gene expression landscape between these 2 populations. And this is exactly what we knew from bulk comparisons of gene expressions as well. And there's also a fair bit of overlap in the middle. So there's kind of this transition from what starts from here and goes there. If you really dig deeper, as I said, a lot of these cell lines are actually quite heterogeneous. There seems to be 2 parental populations here. One of them is not metastatic at all, and it just completely goes away after you select for metastasis. The other one is metastatic, but then you adapt it for higher metastatic capacity. And so you get the green population emerging from this middle purple population. That's kind of the idea. Now, of course, we can't just run 10x on every sample and kind of to make sure that our labels were correct. We have come up with qPCR strategies and things like that over the years to be able to make sure that we have the right cell lines. But this question has always persisted that what else is there that we can actually use, and of course in the context of metastasis, what is really famous is EMT. It's known to be important in metastatic progression. It's epithelial to mesenchymal transition, and it has been known for decades now that it's involved. in metastasis, is actually known to also, cause a lot of morphological changes. So I'm showing you here one of the genes that we identified a few years ago that is a post-transcriptional regulator of EMT, which is really the first post-transcriptional regulator of EMT to be discovered. Before that is just a few transcription factors that were known to be involved in that. And this is an RNA binding protein called RBMS1. And as you can see, if when you knock it down, you get EMT. And the shape of the cells become like elongated. And this is actually one of the few times that you can actually induce EMT, because EMT itself is difficult to induce in culture, but that does happen in vivo again. So when I first heard about Deepcell and looked at their embeddings, and thanks to a friendship with the founders, it pays to be in the Bay Area. So the first thing that popped into my head is that can we actually look at the relationship between, again, we had done a lot of work on morphology and metastasis. We had actually shown that higher variation in morphology is associated with metastasis as a stand-in or proxy for heterogeneity. But kind of, can you functionalize that? I think that, like, the back of my head, the question was basically, can we establish a relationship between function and morphology? That is, if I look like Tom Cruise, which I do, can I act like him? Basically, is the fundamental question. If you accept the premise, I'm happy to start auditioning for roles. I mean, he's retiring from Mission Impossible franchise. The door is wide open for me to walk in. Okay, so this was basically the premise of what we set out to do, and enters Deepcell. As Ken mentioned, we had one of the first, well, not one of the, like the first instrument out of Deepcell's labs, and it looked like this. So it's a prototype. You all saw the REMI one. It looks a lot better, fancier, but I'll take this back anytime if you still have it. So we had a kind of a toy room where we have these kinds of instruments, and actually next to this there's a sequel to PacBio machine. So we were happy to have it for a few months. Unfortunately, got taken away. Then us sobbing didn't help. So hopefully we'll get one back. What was fun, we also got Jan for a few months along with the instrument. We couldn't keep her either. We were happy to, but I don't think she was up for the pay cut that we were going to give her. But you all saw the new instrument, so very much excited about that. So I'm going to tell you a little bit about kind of the vignettes that we worked on, again focusing on the relationship between morphology and function. Some of it is work in progress pretty much. So I can tell you like what we have done so far and how much progress we've made. So the first vignette is, again, I told you about all these models of metastasis. I told you that they look different in 10x, embedding space. Now, do they look different in terms of morphology? Again, emphasis on the fact that most of these don't really look different to us by eye. They, even with microscopy and very quantitative measurements, they don't really look different under microscope either. Some of them do, but most of them don't. So we picked a few of these models, A375. So this is a model of lung cancer metastasis. This is colon cancer metastasis, skin cancer, colon cancer, and these two are breast cancer. So for each of them, we have a parental line and a metastatic line, and we basically did the Deepcell analysis. So the data that's presented basically looks like this. There are two separate clusters because cells are imaged in two flow planes. I don't know if this will persist or not, but basically we collect data from the same cells two ways. So each of them, you can see that the parentals and the highly metastatic derivatives are colored separately, and lo and behold, you actually start to see the differences. And sometimes some samples fail. I mean, some cell lines fail, but there are quite a few success stories. Again, to my eye, these two never look different. So this is big accomplishment from my perspective, that you can actually start to see that differences emerge. So for example, this is a density plots. You're now at this point familiar with these, so I don't spend a lot of time describing them, but basically you go from like the parental line here, to more here and down there for the highly metastatic cells. And it's even better in this flow plane. There's a very clear movement of cells in the embedding space. And again, similar to the previous slides you saw, you can also look at confusion matrices. Now the idea is that can we use these morphometric features that the models have been trained on to actually classify a cell. So if I see a cell, can I tell if it's highly metastatic or not just based on its morphological feature? It does pretty well. Again, really emphasis on the fact that these cells are not that very different. And even in single-cell RNA-seq, we see quite a bit of overlap. So even like single-cell RNA-seq would not look that much better than this confusion matrix for this cell line model. So in terms of future directions that we need to do for this vignette, I would say, we want to go back to that idea of cancer cell heterogeneity, right? So the idea that I told you about was that, there are two populations of this line. One of them dies, the other one survives and is metastatic. So this is all like gibberish. We haven't actually shown this. The way to show this is to do like lineage tracing, where you find barcodes and fish out those barcodes, the cells with those barcodes, and then go and do assays, which I haven't really been able to convince any graduate students to do yet. But here we can basically go back and sort that population specifically and ask whether this population that looks like metastatic cells, is it actually more metastatic? Again, going back to the morphology versus function. So if I pick a cell that looks metastatic, will it also be more metastatic in mouse models? So of course we still have to do that experiment, but the initial data looks very promising. 50,000 is usually the number of cells we need. We can even go lower. 50,000 is actually the max because we want to follow them for a few weeks. And these models specifically are quite metastatic. So this was one of the vignettes. If you have any questions about this part, okay.
Hani, do you see those two subpopulations of parental also in morphology? Like if you go back to those, there was an upper tiny lobe, but that's also shared between parental and metastatic.
Yeah, exactly. So the point that I was trying to make is that there are parental lines here, right? That are residing in the same space as the highly metastatic ones. So if I pick these parentals, will they be compared to relative to these parentals where highly metastatic cells are not? Will I see like a separation in phenotype? That's kind of our expectation.
What about looking at like if you have a bank with known outcomes,
Mm-hmm.
of primary tumors from patients that went on to be metastatic? Do you think there's any morphological information in the primary tumor that can predict?
I would say absolutely, because the way that we really think about metastasis is that it's not something that you kind of evolve, like a cell does not start on its journey and adapt over time. The idea is that there is a population, a subpopulation in the primary tumor that can do this and eventually does this. So that's why like having a higher heterogeneity helps. There's just more shots on goal, basically. So the idea is that there is a subpopulation, and now if that subpopulation has different morphological properties, like we were kind of making the case for here, then if you see those, then it means that this tumor is more metastatic. And we have made this kind of observation again in the context of single cell sequencing, for example. You can find rare but what they call cancer stem cells, for example, right? But also like cells that have like ENT signatures and things like that, and they're very much associated with, later metastasis. And all of that signal is basically gone when you look at the bulk, of course, so you need that kind of single cell resolution.
The question I have, what is the rarity level that you can detect? And how many cells do you actually need to be able to, you're talking about a very minor population of cells in the primary tumor, right?
I mean, with 10x, you still do like 10,000 per sample usually. I mean, you can go higher, but most folks do in that number. But yeah, I think we want to see at least things like 10, 50 times. That's kind of our go-to.
So the answer to that question really depends looking at supervised models, we had applications where our false positive rate went down to 10 to the negative 5. But we were looking for like uncommon cancer cells in blood, like in the CTC setting, kind of an application. And you have a huge morphological difference between your background cells, which are immune, and your target cells, so you can't go that low. Here, you need to see whether that subpopulation pops out.
Yeah, exactly. So these are all the same cell type, even cell subtype, and so it's the differences are way more nuanced than where you have like a cancer cell, which is basically an epithelial cell in blood versus other immune cells. Okay, so the second snippet, in cancer biology, in addition to metastasis, you also do a lot of drug work, right? So we care about resistance, we care about basically kind of how cells survive, like mechanism of action of the drugs, and mechanisms of tolerance, basically. So one of the questions that we also wanted to ask early on was that, do we actually see morphological differences for cells that are treated with different drugs? We specifically picked Decitabine because it's a methyltransferase inhibitor, so you basically get like a massive change in the gene expression landscape. Now the question is, does that change also reflect changes in morphology? It actually does a fair bit, as you can see. So they go from here to there. So these are happily growing cells. These are dying cells. They also look funky. I think that's the best way I can describe them. They kind of have this like jagged surface around the cell. We associate these cells with like dying cells, but they can actually persist quite a while. But they're kind of on their way to death, but they persist quite a bit. And again, this might be even an easier task, as you can imagine, from those images that you saw earlier. We can classify quite easily which cells are treated and which cells are untreated, especially at the higher concentration, which these results are from. And you can, again, what is nice about the morphometric classifiers is that you can go back and look at different features that went into this classifier and ask like what was important. Again, size and texture was important, and those were actually obvious from the images themselves. So we expanded on this. So there are a couple of ways that we want to kind of think about drug treatment, right? So one is that, well, we made this observation that you dump some compound on a cell and the cell changes, right? So, but in order to make this really useful, you need to show that if I add different drugs, the cell changes in different ways, right? So that's one observation, because different drugs have different mechanisms of action. And more importantly, if I add different drugs of the same mechanism of action, then they again look similar. So these are the two things that we need to think about. Again, we are halfway through this. So the first part of it we have done. So going back to kind of the study plan, we basically added more resolution. We took multiple time points with the treatments. We also added another drug, which is an EZH2 inhibitor. So basically, we have more time points. It's day 1, day 3, day 5, and day 8. And I'm showing you some of them here. Basically, I don't know if you can read them, but 1, 3, 5, 8. We have control samples, Decitabine, and this is our EZH2 inhibitor, tazemetostat. So again, you can see like shifts in the morphology, which is what you'd expect. Control cells just happily reside here. This seems to be the happy place, the good place. And then for both treatments, cells end up in this final state of like what we call the death black hole. I'm sure there's kind of a philosophical message there. In death, all cells look alike, I think is the lesson here. But how they get there is actually quite different for different drugs. And this is a really good thing because you want them to be, right? And again, you can go back and look at different parts of this embedding space and see what the cells look like. So this is our EZH2 inhibitor. This is the final day again. Like you can see in the dying population, you can see the smaller size and the jaggedy rough surface. But you can also look at different parts of this. So this is definitely a kind of a resistance pocket, right? So these are cells that should be dying, but they're not. They actually look pretty happy, and this is your resistance pocket, or same here, right? So going back the same way that we were talking about how finding subpopulations of cells that look metastatic, then it means that the patient will do poorly. The same applies here. The cells that look like they should be tolerant, they could actually be tolerant, and that's bad news for the patient.
I mean, are these cells, are you going to characterize them in some molecular way and find out, for example, they've got some kind of membrane pump that's pumping these drugs out actively? I know there's the whole side population thing with cancer stem cells that has some debate around it, but quite interesting.
Yeah, so mechanisms of tolerance here are actually complicated for these kind of drugs that hit epigenetics, basically. So it's from like downstream effects that try to counteract it. So you're hitting an HDAC, and then you get a DAC upregulation, things like that. But some of it is also this kind of just this broad efflux pumps. But they are very much insufficient to explain the kind of variation that you see, the heterogeneity that you see in the population. They are really a big problem in the field, but there are general mechanism of tolerance, but there is a ton of specific mechanisms of tolerance as well. And so some of these states could be basically reflecting those mechanisms. So that was, as you saw, this was our EZH2 inhibitor. This one is Decitabine. Again, it looks decidedly different, right? So we used two different drugs, two mechanisms of action, and they look very different on their way to death. And again, in death, they look similar, so which is what we expect. Now, as I mentioned, this is, again, preliminary. What we need to show is that if we then take another EZH2 inhibitor, then it will look like this, right? So if basically we can classify drugs based on their mechanism of action and overlay it on top of this trajectory that they take over time towards cell death, right? So if we can do that, then we can go the opposite, where we take a drug that we don't know how it functions and then match it to one of these trajectories, okay? And that would be your, it can be either understanding a mechanism of action for a drug that you just happen to find through a phenotypic screen of sorts, or, a drug that no one has ever seen before. It's a new, it's basically an NCE that you want to test and figure out how it functions. Okay, so these are kind of a summary of the observations that we made. As I mentioned, we do see morphological changes. That is expected. We do see different ones for different drugs based on their mechanism of action, which is pretty good. Again, remains to be seen if we can take drugs from the same mechanism and show that they look similar. And you can actually make really good observations around. I didn't spend a lot of time on this. Like, the data is just so rich. Again, once you get to this high dimensional data, you can spend 5 years on them, unfortunately. Similar to single cell sequencing, but you can actually peer into each of these clusters. And again, the idea being that if you can sort out different parts of this population, so for example, if we go here, I said these are likely resistant. So again, if you go to the parental population and fish out this part of the embedding space, they should actually be more tolerant. So are they? So that remains to be seen. Okay, so this was the second vignette. Any questions here?
Yes. You go back to the images that you just had. So one more. So the center population is really interesting. How interpretable, this might be a naive question, but how interpretable are the images that we're looking at right now to the human eye? This is more like a cytology question. Relative to the data that we have, or the potential molecular data that you could generate?
So this one, so these ones actually are quite different. So there are cases in which they're not, but you can go back to the model and see which which of the metrics are important for it. And sometimes it's like many of them that have to be together, and that's why like machine learning is so powerful in the end, like nonlinear interactions between features basically. But these ones specifically are obvious actually. If you're a little bit far, but if you look at them, these are like the texture is very different here from here. And these are very circular, actually almost too circular. Whereas these guys look more normal, and some of them are doublets and things like that.
Some of the top features of importance were more the orthogonal, orthogonal features. Could you go back and reanalyze this data just using those, or removing some of the maybe size-associated ones to dig deeper into the impact of the different drugs?
I don't know how, maybe Deepcell folks can tell us how easy it is to like turn off different features or not use them or things like that in their model.
So you would have access to your own embedding data set. So the 115 dimensions of the embedding, you have access to that. But obviously the model that created that embedding is relying on all the pixels, right? You can look at a specific dimension in one of those 115, and then we do have the description of which ones are morphometric, which ones are AI driven. So you can do all sorts of analysis to look at every individual or remove individuals and then create a UMAP or whatever visualization you want based off of that. On the Axon data suite, we also have an ability to just turn on one at a time and look at a heat map of each individual parameter to see what parts of the map Axon is kind of like maybe focusing more on. But like all sorts of like interpretability questions, visualization questions, we're really excited to hear more and provide as much richness to the visualization as we can. The fundamental of like just the raw data should give you what you need, I think, with your current workflows.
I was most interested in the last bit that you said, that the potential to then go and sort of look at specific subpopulations that may be of interest. So can you tell me what would be such a potential follow-up that you might do with a study like this? So you've got these drugs, you're seeing what are the heterogeneity of response, and you're seeing the rescue and stuff. But then, what is the kind of sorting you wish you would potentially do next?
So yeah, so this is, for example, this is a treated population, right? And from it, you basically hypothesize that these are pockets of resistance. So if you go to their parental population, I don't know if you go here, right, there are cells that like reside there to begin with, because they're a minority, right? And those are the ones that you want to sort out and then ask, are they more resistant? Right. If they are, then basically you have linked how the cells look as a minority in the heterogeneous population to their ability to tolerate the drug. So they're predefined pockets of resistance in your population. If they do exist, it means that you can find them. So in a clinical sample, for example, you can actually predict who will respond and who will not based on the heterogeneity that you see in the population.
Just a quick question, Doc. With the mode of action of these drugs, do they require the cells to be actively proliferating?
Decitabine definitely does. EZH2 probably does as well, yeah.
Because there is actually, in all the cell lines, there's a strange population of cells that don't die, that don't proliferate, they just sit there.
Yeah, so we specifically picked these drugs in this cell line, K562, because they actually die quite happily. I mean, these are the dead cells, as I said, right? So this cell line is sensitive to these drugs. But it's also be interesting to check cell lines that are resistant and then see what happens to the morphology in that context, if anything at all. I mean, as I said, these are all like vignettes, so there's a lot that can be done, either on like sensitive lines or resistant lines, different drug classes and things like that. But just more like, these are pilots, proof of concepts. Is there anything to be explored here? And short answer for me is definitely. So the final vignette, quickly, I know we went over time a little bit. So we also did while we had the instrument, we also did a large clinical study. It was kind of timely. We were doing our own thing, and I think Deepcell was doing something similar, so we kind of joined forces. And this is basically looking at malignant pleural effusions and ascites in patients. These are actually quite common in late-stage cancers, especially in breast cancer and lung cancer. And as I mentioned, a lot of our cancer cell line models actually are derived from these pleural effusions. So you collect them from patients. So it's basically fluid buildup either in the chest or abdomen, and you actually have to empty it periodically. And so you get like a ton of liquid. And in that liquid, there are a ton of cells. And some of them are cancer cells, and some of them are immune cells and other cell types. But one of the questions that one of our collaborators, Mark, had was basically how can we take advantage of these fluids that, for the most part, folks are throwing away to basically start to study the cancer by focusing on that minute population that is cancer cells. So this is actually the general kind of proportion of cell types that you see. And again, these are the cancer cells, the absolute minority. So actually fishing this out is quite a lot of work because you get a ton of fluid, and you have to run sorting it is a whole process. So we were actually happy to join forces with Deepcell to do this. So this is the workflow. Our samples came in, and so part of it went through Deepcell's platform, and we got cancer cells sorted out and done low-pass genome sequencing, whole genome sequencing. We also did 10x single-cell RNA sequencing on the whole population. We are still analyzing that data, so I'm not including it here, but I would say the fraction of the population that was, for each sample, that was called as cancer cells by Deepcell was very much in agreement in what we were calling as cancer cells and 10x data. So again, there's a very, very good concordance there, which is expected. So this was kind of a tour de force. We had 10 patients, a variety of sample sizes per patient. And so overall, 26 samples. This is all thanks to Mark and my lab. He was very good at, leading everyone into battle. It took a while to collect all of these samples, but it has been kind of a very useful resource so far. So a few interesting kind of observations. Again, as I mentioned, there is general concordance between our analysis with other modalities and the predictions from Deepcell, that what fraction of cells are cancer. As I mentioned, we have also done 10x, which is not here, but also agrees with these. And more importantly, post-sort, we can also enrich for the cancer cells, and this becomes quite valuable, I would say, because this allows us to do the kinds of assays, be it functional assays or even more deeper molecular assays. So with 10x, you can only get the transcriptome, and you basically kill the cell in the process anyways. You get this low quality data. But with this approach, we'll be able to collect cancer cells, do RiboSeq that we love to do IsoSeq that we love to do, kind of do much deeper kind of molecular profiling of cancer cells than single-cell genomics allows us to do. And more importantly, even do functional assays, put them back in mice, even grow them into PDX models and things like that. So one thing that we have noted so far based on just the morphometric features of putting all the cancer cells together is that, again, there's a fair bit of heterogeneity. This is expected, and it's a good thing to see. That different cancer cells look different. They're all very much distinguishable from all the other cells that are there, but they're also distinguishable from each other. So you're not only simply capturing the fact that these are epithelial cells, you're actually learning something more about individual cancer cells. And if you look at these patient samples, you basically as I said, we did low-pass copy number analysis, and you actually get this, the kind of amplifications that you would expect, gains and deletions. There are a couple of very famous amplifications that we have found, and more importantly, these are again enriched very strongly post-sort. So this is very good to see because, again, these are actually difficult to this is a good sample, and you see there's just a tiny bump. Whereas post-sort, we can very strongly enrich for that signal. And more importantly, there's also, we already heard about these kinds of applications, but there's also room for thinking about kind of like liquid biopsy approaches to this modality, right? Kind of, you can, for the samples that we had multiple time points, you can actually see how the tumor cells behave, as the patient is being treated. So some of them kind of go away, the tumor cells, some of them go away and then come back, which doesn't bode well for the patient. And also, again, you can go back to the genetic analysis and that we have done on these cells and see if there is association between how they look and their genetic backgrounds and genetic drivers. We haven't really done that analysis. It's still work in progress, but it's something that we can actually do. But of course, you need a lot more samples to be able to say, for example, P53 like null cells tend to look this way, or like ESR1 mutant cells tend to look this other way. But again, this requires this kind of bringing of the two worlds together, where you do this molecular phenotyping and put it together with morphometric phenotyping and kind of learn a joint embedding of the data, basically, is the point. But what we can really do effectively, we have already demonstrated, is that you can actually do this patient tracking. As I said, for one of the patients, we had multiple samples, so you can actually look at how the tumor responds and evolves over time just by simply following that population in this sea of like material and other cell types. So again, this was a more run-of-the-mill vignette that I think you have heard examples of, and hopefully you'll hear more. But it has been very enabling for us. As I said, it's very difficult to collect cancer cells from these pleural effusion kind of samples that we collect. So it can be actually quite enabling in the clinic. So with that, I'm going to thank the folks who actually did this. Of course, Deepcell folks who gave us the instrument, and Jen. We'll have her back, whenever you're ready. And also the folks in my lab who did the work. A lot of the cell line work was basically done by Bahar and Timmy in the lab. And our techs, all of them were involved in the pleural effusion sample collection. And also Xiaopu, the postdoc in my lab, did all the 10x, which I haven't presented yet. And I'm happy to take any more questions you might have.